morning, everybody, and welcome to this session. And I'll be speaking about the influence of genes on myopia. This is the most exciting field right now in myopia. Uh, if you sleep through this talk, you risk becoming a dinosaur. So why I'm telling you this is because in the graph you are seeing on the screen, you see the number of genes associated with myopia has just grown exponentially in the last 5 years. In the up to the year 2015, 2018, there were around 20 genes known to exist which were associated with myopia. Right now there are 450 genes. And I am going to make a strong prediction today that in the next 10 years we will come to a point where the first test you will do for a myopic child is his genetic profile. And only then you will start any treatment or intervention. And this we will discuss again after 10 years and you will say I was right. So please stay awake. Now, this slide is not moving. My slide has slipped. Okay. So why do we think that there is a genetic basis for myopia? We all know that we always, without even knowing genetics, we know that if the parents have myopia, the children are likely to get myopia. So there is a familial clustering of myopia. If one twin, monozygotic twin has myopia, the other one is very likely to have myopia, which is not true for a dizygotic twin. And then there are syndromes which are strongly associated with myopia. You have Strickler's syndrome, Marfan syndrome. So these are definitely genetic diseases which have myopia. So maybe myopia has a genetic basis. And there are myopia associated with other heritable eye conditions like congenital stationary blindness, achromatopsia, and others, where again there is a strong genetic basis. So you think that myopia may have a genetic basis. Now the older studies were based on these kind of observations where you saw clustering of myopia in families or twins with myopia, and then you extracted their DNA and you tried to find what was common in them by doing something called as a linkage analysis, which is a statistical tool to find out what the similarities between two DNAs. And then these genes, these studies were able to find out genes which were pretty large, and because they were these loci were loci is the section in the chromosome, and every loci will have several genes. So these studies actually identified the loci. Okay, where in if suppose the DNA strand is like a building, and there are 23 buildings, and each building has got a flat, and there are several flats. So the buildings will be the loci, and the flats will be the genes. So it's like that. So these studies, the earlier studies were actually able to tell you, okay, in this A wing, but they did not know which flat, so which gene is there, they didn't know. Now, because these were large loci, it was very difficult to tell which part of the loci was problematic. So the first gene was discovered in 1990, and then from then on, these are the genes associated with non-syndromic myopia and not with syndromic myopia. So you have the genes ranging from myopia 1 gene to myopia 27, but there are in all total 26 genes right now which are known to be associated with non-syndromic myopia. Out of this, most of the genes are associated with high myopia, and only 6 genes are found to be associated with what you call as school myopia or common myopia, which are gene number myopia 6, 7, 8, 9, 10, and 14. And the remaining ones are associated with high myopia. Now, these were all a product of linkage analysis. So you have to understand how genetics works. So the first myopia gene was discovered in 1990, but myopia being an inherited disease was first discovered in 1980. So there was a Danish family which was having three brothers, and they had 5 grandchildren who all were having high myopia. So they postulated that this is a X-linked recessive gene. And then it took 10 years for them to find that it was the XQ28 loci, which was actually responsible for this kind of X-linked inheritance. 20 years to find another family to have the similar gene. And then it took another 5 years to find the actual gene in that particular strand. So you'd see from the time that X-linked recessive myopia was discovered till the discovery of the gene, it took almost 35 years. Now, mechanism is still not identified. They have identified the gene, but how the gene causes myopia, we still don't know. That might take another 10 years. But it is associated with both syndromic and non-syndromic. This is just an example I am giving, and this is true for all the other 27 genes. Now we will come to something called as candidate gene. Now in these loci, they have certain known candidate genes. These genes are having known biological functions. As you know that the human genome has 6,000 genes which are known to have some biological function. So when you find these loci, you try to see which are the active genes or that cause some problem or some function in a pathway, and then you try to test whether this gene can be associated with causation of myopia. So they have tested all these collagen 1, collagen 2, transforming growth factor gene, lumican, myocillin, PAC6 genes, and all of these has been tested as candidate genes postulated being responsible for causing myopia. And although some studies say that they are responsible, they are not replicated in other studies. Replication in other studies is very important significance, because then it actually proves that your postulation is right. So only 7 candidate genes and mutations have been replicated so far amongst all these 27 genes. These loci have a strong inheritance effects, and most of them are autosomal dominant, only 2 of them are autosomal recessive. And because when they are inherited in a Mendelian fashion, and these are large loci, they cause high myopia. So very few of them actually explain your common simple myopia. Most of them explain non-syndromic high myopia. Now we come to the newer, most exciting field in genetics right now, which is called genome-wide association studies. Now, genome-wide association studies is data mining of genomic banks. So if you have large number of people and you collect their genes, these are unrelated people. These are not from the same family. These live in the same area. And then you have collected their DNA, and then you see, okay, amongst these, how many people have myopia and how many people don't have myopia. And then I try to find the common genes in the people who have myopia and their probability of being associated with that particular trait, that being myopia or axial length or corneal curvature or refractive error. So you take this to a deep data mining, you can see. You can go on digging this with supercomputers. You can read the, common traits. But the only drawback is that you are able to only find one kind of mutation in these studies, which is called single nucleotide polymorphisms, which is replacement of one nucleotide in that particular gene. Now, we have identified around 400 SNPs in adult myopia so far. Now, how does the output of GWS looks like? It is like a Manhattan skyline. So you have all these chromosomes from 1 to 23 in these colored bands, and the towers that you are seeing are the SNPs in the different loci. Now, only the SNPs which reach a significant of 1 in a billion is taken to be significant. That means you will have so many associated genes, literally in thousands, and the only ones you pick are the ones which have a probability of less than 1 into 10 raised to minus 8 to happen in that subset. That is the amount of significance you want to have. This is to remove the noise from the signal. I'll need 2-3 minutes more because it's a very difficult topic. I can't finish in 8 minutes. Okay. So these SNPs are, because they are only single nucleotide morphisms, that means replacement of one nucleotide with another, they do not have a profound effect on the corresponding protein. That is why they will not have a large effect size. So maybe one SNP will have effect of say 0.01 diopter of myopia. And that's why a person with common myopia will have hundreds and thousands of these SNPs. And the sum total of that will lead to that myopia of 4 diopters or 3 diopters. Now, only few SNPs get replicated in other loci, like 15q14 and 15q25 are the most replicated ones. Now, where are these genes expressed? So these genes are expressed in all layers of the retina, right? And in the sclera also. So that's why the postulate is that the theory that we proved with animal studies, that it is a local phenomenon where the defocus on the retina causes the elongation of the eyeball. That is proved by these genes, the location of these, the expression of these genes. Now we come to how will you use this in practice. So you calculate something called as the polygenic risk score for predicting myopia. So you do a DNA profile of a person, you see how many of these SNPs he has, you see the total of that, and you predict whether this person is likely to get myopia or not. So right now also there are several PRS available, but none of them is strong enough to explain all of common myopia. That means thousands of more SNPs remain to be discovered. Now the gene environment interaction is very important because in certain genes require environmental influence to express themselves. As we know, people who go outdoors have less myopia, people who study more have more myopia. That means these environmental influences affect the expression of the genes. And there have been studies for this, and they have found that in Singapore, they studied these SNPs, and they found that the association of the SNPs with which were responsible for higher education were also associated with SNPs responsible for myopia. So that means the environmental influences also have a bearing on the expression of the gene. It also affects the effect of treatment on a person. Like you put atropine to so many people, but there are non-responders. So that is based on the genes. So there was a study done in Spain where they found that the effect of myosite lenses on people with certain genes was much more than the people who did not have that gene. So these are the kind of studies that are happening. The future directions are more studies with whole exome sequence and whole genome sequence to look at rare variants and other mutations like copy number variations have to be done, because we are looking at only one kind of mutation. The other kind of mutation may also be contributing to the whole effect. And a robust risk prediction based on genetic profile is the future. Thank you very much.
Thank you, sir. Thank you, Dr. Siddharth. I think it was very exhaustive and a much needed coverage on genetics.
Yeah, no, that's one of the best genetic talks I've heard on myopia. It was very succinct.
Yes.
And if you were sleeping through it, you missed it.
Absolutely.
That was like taking a capsule of all the information. That's a lot of hours of work.
Yes.
Congratulations.
Yes. So, yeah, I think a big round of applause for Dr. Siddharth.